DNA, deoxyribonucleic acid, is a molecule that carries the genetic instructions for the development, function, and reproduction of living organisms, sorted as a sequence of 4 chemical bases, adenine pairing with thymine and guanine pairing with cytosine, that are arranged in a double helix. When the chemical base pairs pair up with their complementary base pair, they form hydrogen bonds to create the stable structure of DNA double helix. The pairing is particularly important to maintain the structure and accurate DNA replication and inheritance of genetic information. Addition of a methyl group to DNA, usually at cytosine bases that are part of CpG islands, which are regions with a high frequency of cytosine and guanine nucleotides linked by phosphate groups often located near gene promoter regions. Specific sequences of DNA located near the start of a gene act like on and off switches for transcription. They are essential sites where RNA polymerase and transcription factors bind to initiate the process of copying DNA into RNA. Methylation can prevent transcription factors from binding to DNA, effectively silencing gene expression. This process plays a critical role in epigenetic regulation because it allows cells to turn genes on or off without altering the underlying DNA sequence, influencing development and risks of diseases like cancer. Acetylation involves the addition of an acetyl group to histone proteins, which causes the chromatin structure to loosen. This loosening allows transcription enzymes like RNA polymerase easier access to the DNA, thereby activating gene expression. The balance between acetylation and deacetylation is essential for regulating efficient transcription in cells. Proto-oncogenes are mutated or overexpressed versions of proto-oncogenes, which are mutated or overexpressed versions of proto-oncogenes that are normal genes regulating cell growth, division, and differentiation to ensure tissue development and function properly. When proto-oncogenes become oncogenes, they can drive uncontrolled cell proliferation, which may lead to cancer. These changes can result from point mutations such as single base substitution. Several types of mutations can affect DNA sequences, which will alter the way that certain genes are expressed. For example, point mutations involve a change in a single base and can be silent, missense, or nonsense. Insertions or deletions, also called indels, can cause frameshift mutations. Other types include duplications where segments of DNA are copied, inversions where a segment is flipped in orientation, and translocation where a DNA segment moves to a different chromosome. Mutations can occur in somatic cells, which are non-heritable, or germline cells, which can be passed on to offspring. Introns and exons. These are genes in eukaryotic cells. Introns are non-coding sequences with genes that are transcribed into RNA but removed during RNA splicing, which occurs before the RNA leaves the nucleus. Although introns are not translated into proteins, they can contain regulatory elements that influence how genes are expressed. In contrast, exons are the coding sequences of a gene that are retained in mature mRNA and translated into protein. This splicing process is important because it allows alternative splicing, where exons can be combined in different ways to produce different protein variants from a single gene, increasing the diversity of proteins a cell can make. Polymerase chain reaction, or PCR, is a laboratory technique used to amplify specific DNA sequences, producing millions of copies from a small initial sample. The process requires the enzyme DNA polymerase, commonly Taq polymerase, and occurs through repeated heated and cooling annealing cycles. During denaturation at around 95 degrees Celsius, the double-stranded DNA separates into single strands. During annealing at 50 to 65 degrees Celsius, short primers bind to complementary sequences on the DNA template. During extension at 72 degrees Celsius approximately, DNA polymerase synthesizes new DNA strands by adding nucleotides. PCR is essential in diagnosis, forensic analysis, and genetic research because it allows tiny amounts of DNA to be studied in detail. CRISPR-Cas9 is a genome editing tool derived from bacterial immune systems. It uses a guide RNA to direct the Cas9 enzyme to a specific DNA sequence where Cas9 introduces a precise double-stranded cut. This allows genes to be knocked out, replaced, or modified, enabling applications such as gene therapy, disease modeling, and improvements in agriculture. CRISPR's precision and versatility make it a powerful tool for research and real-world biotechnology applications. Gel electrophoresis is a technique used to separate DNA, RNA, or proteins based on their size and charge. DNA samples are loaded onto an agarose gel, and an electric current moves the negatively charged DNA fragments toward the positive electrode. Smaller fragments move faster, while larger fragments move more slowly. Gel electrophoresis is commonly used after PCR to visualize DNA fragments check for successful amplification, and compare fragment sizes in genetic analysis or forensic testing.
DNA stores and transmits genetic information, serving as the universal code for life across all organisms. It is organized into chromosomes, which are made up of genes, specific sequences of nucleotides that code for functional protein or RNA molecules. While this function is shared by all life, the way DNA is structured differs between prokaryotes and eukaryotes. Prokaryotes, the earliest and simplest organisms, have DNA that is unbound, circular, and double stranded, located in the cytosol within the nucleoid region. Although they lack histones, they do contain protein that help organize their genetic material. In contrast, eukaryotes have DNA enclosed within nucleus, packaged into multiple linear chromosomes. Their DNA is tightly wound around histone proteins, forming chromatin that allows for efficient organization and regulation. Additionally, plants and some other eukaryotic cells stores DNA in chloroplast and mitochondria. This structure versatility ensures DNA remains protected while enabling its vital role in inheritance and function. Enzymes are globular protein molecules that act as biological catalysts, speeding up specific chemical reactions essential for life. Their unique structure gives them specificity. Each enzyme has an active site shaped to bind a particular substrate. The induced Fist model explains how the active site molds around the substrate, forming an enzyme-substrate complex that places bonds under stress and lowers the activation energy needed for the reaction. This enables reaction to occur much faster without enzyme being consumed. However, enzymes are highly sensitive to their environment. Extreme temperature or pH can disrupt the protein's folding and hydrogen bonds, altering the active site. Once denatured, the enzyme and DNA polymerase builds DNA strands. Tip: if a biological molecule ends with ase, it is usually an enzyme. Mitosis is the process by which an eukaryotic cell divides to produce two genetically identical daughter cells, ensuring growth, repair, and maintenance of tissue. It is a highly organized sequence of stages that preserves the chromosome number from one generation of cells to the next. In prophase, chromosomes condense and become visible. While the nuclear membrane begins to break down, metaphase aligns the chromosomes along the equator of the cell attached to the spindle fibers. Despite anaphase, sister chromatids are pulled apart to the opposite poles, ensuring each new cell receives an identical set of chromosomes. Finally, in telophase, nuclear membranes reform around to ensuring each new cell receives an identical set of chromosomes. Finally, in telophase, nuclear membranes reform around two sets of chromosomes, followed by cytokinesis, which divides the cytoplasm. Mitosis is tightly regulated as errors can lead to mutations or uncontrolled cell growth, such as cancer. This precise process highlights how cells maintain genetic stability while supporting life's continuity. Tips: vocabulary to use in your exam. DNA structure and function. DNA, the molecule that stores genetic information. Nucleotide, building block of DNA made of sugar, a phosphate group, and nitrogenous base. Nitrogenous bases: adenine A, thymine T, cytosine C, guanine G. Complementary base pairing: A pairs with T, C pairs with G. Double helix, twisted ladder like structure of DNA. Chromosome, a long thread of DNA wound around proteins. Gene, a sequence of DNA that codes for a protein or RNA molecule. Histones, protein that DNA wraps around for organization. Replication, the process of copying DNA before cell division. Tips: vocabulary to use in your exams. Enzymes. Enzyme, a biological catalyst made of proteins that speed up the reaction without being used up. Active site, the pocket or groove on an enzyme where substrate binds. Substrate, the molecule that enzymes act upon. Substrate enzyme complex, the temporary structure formed when substrate bind to an enzyme. Induced fit model theory that enzyme's active site slightly changes shape to fit the substrate more closely. Activation energy, the minimum energy needed for a reaction, enzymes lower this. Denature, the loss of an enzyme shape and function due to extreme temperature or pH. Specificity, the property that each enzyme usually acts upon only one substrate. Mitosis, cell division producing two genetically identical daughter cells. Prophase, chromosomes contents, nuclear membrane breakdowns. Metaphase, chromosomes line up at the equator of the cell. Anaphase, sister chromatids are pulled apart to opposite poles. Telophase, nuclear membranes reform around chromosomes. Cytokinesis, cytoplasm divides, completing cell division. Spindle fibers, structures that pull chromatids apart. Chromatid, one half of a duplicated chromosome. Genetic stability. The accuracy of mitosis ensures identical genetic material in daughter cells.
DNA replication is the process that occurs before the cell divides, as it needs to make an exact copy of the DNA so that both new cells receive the same instructions. The DNA double helix unwinds enzymes like helicase, separates the strands, and DNA polymerase builds new complementary strands, resulting in two identical DNA molecules, and that marks the foundation for everything else. Transcription and translation are among the most misunderstood processes. Transcription, the first step in making proteins, is like a recipe from the book. The enzyme RNA polymerase reads a section of DNA and makes a single-stranded copy called messenger RNA or mRNA. This mRNA leaves the nucleus and carries the instructions out into the cytoplasm. Translation is the second and final step. It is when the cell utilizes the protein, like using that recipe to cook. The mRNA travels to a ribosome, which reads a genetic code in groups of 3 bases called codons. Each codon matches up with a transfer RNA or tRNA carrying a specific amino acid. As the ribosome moves along the mRNA, the amino acids link together in the correct order, forming a protein. So transcription writes the message and translation reads it to make the product. Meiosis is not about making protein, but rather making gametes like sperm and eggs. Instead of producing identical cells, meiosis cuts the chromosome number in half. Humans normally have 46 chromosomes, but gametes only get 23. Before meiosis begins, the DNA is replicated, so each chromosome has 2 identical sister chromatids. Then comes meiosis. In this stage, homologous chromosomes, one from mom and one from dad, pair up. They can swap sections of DNA in a process called crossing over, which increases genetic variation. After that, the pairs are pulled apart into 2 separate cells. Each of those cells now has half the number of chromosomes, but the chromosomes are still doubled as sister chromatids. Following on is meiosis 2. The sister chromatids are separated and pulled to opposite ends of the cell, where the cell divides again. It results in 4 daughter cells. Each one has 23 single chromosomes, not 46. Each cell is genetically unique because of crossing over and independent assortment. That's why siblings can look so different, resulting in every gamete and every new individual being one of a kind.